Hello everyone. So today we are also going to continue chapter number 5, that is molecular basis of inheritance, and this is theory part 8, and in this we are going to cover the NCERT section 5.6, 5.6.1 and 5.6.2. So basically today what we are going to do, today we are going to learn more about the genetic code, like how from that mRNA this protein is going to be translated, means that mRNA going to be translated to protein. So let's start. Disclaimer. So what are going to be today's learning objective? First we will understand the triplet codon system. So in this we are going to understand how this codon system, how this triplet codon is going to code for one amino acid, its property and many more things. Then we will learn about this genetic code properties, like how they are universal, they are non-overlapping, but they are degenerate. Everything we are going to understand in today's class. Then we will recognize the different types of mutation. That is what? That is point mutation and frame shift mutation. In today's mutation, we are going to talk about these two mutations only. Then we will understand the structure and function of tRNA. So from last few classes we are talking about this tRNA, which is a transfer RNA, that how it brings the amino acid to ribosome and all those things. But what is its structure and how it do it function, that we are going to understand in today's class. Then what is going to be today's index? So first we are going to cover the NCERT section 5.6, which is genetic code, and in that we have two subsection, that is mutation and genetic code, as well as tRNA, the adapter molecule, that we are going to focus in today's class. Then there will be a summary in which we are going to talk about whatever we are going to study in today's class. We are going to sum up it all in last summary. Then there will be some NCERT based questions that we guys gonna solve together. Then there are some keynotes followed by interesting questions, that are the questions that you guys gonna answer yourself. So here let's come to the topic. So genetic code. What does this genetic code? So first we have to understand this amino acid gap. So we know that the replication and transcription are relatively straightforward due to the principle of complementarity between nucleic acid. So even in case of replication, we know that what happened. So one is the parental strand and the other strand will be newly synthesized strand. On which basis? Complementary basis. We know that purine is going to complement with pyrimidine. Like adenine is going to bind with thymine. Guanine is going to bind with cytosine. And whereas in transcription, we know it follows the same principle of complementarity. The only difference is that the adenine is going to complement with whom? Adenine is going to complement with uracil, and guanine is going to complement with cytosine. So this is very straightforward. But we know that translation do what? Translation converts nucleotide polymers, that is the nucleic acid, into amino acid polymers, which are what? Which are protein. We know that this we talked about replication, then we talk about the transcription. In both the case, what we are using? We are using the principle of complementarity, which is very simple, that purine is going to complement with pyrimidine. But in translation, what is happening? We know that mRNA is translating into what? Is translating into proteins. So what actually is converting? This nucleotide polymer, which is the nucleic acid, is going to convert into amino acid polymers, which is what? Which is protein. But there is no direct complementarity which exists between these nucleotides and amino acid. Again, I am saying that in replication and transcription, we know that there is this complementarity rule where purine is going to complement with pyrimidine. But in this translation step, when the nucleotide polymer, when the mRNA is going to convert into amino acid polymers, that is protein, there is no direct complementarity. We don't know any till now complementarity, till now as in this chapter, till this portion, we don't know any complementarity that is existing between nucleotides and amino acids. Changes in the nucleic acid influence the amino acid sequence in the protein. But we know that if there is some changes in this nucleic acid, it is going to affect the amino acid sequence in where? In proteins. Those concepts led to the idea of a genetic code. So now scientists have started thinking like how it is possible, like we don't know any complementarity rule that exists between the nucleotide and amino acid, but we know that if there is any change in this nucleic acid, it's going to influence the amino acid sequence in protein. So how is it been possible? So this concept led to the idea of what? Of genetic code, a system that dictates the amino acid sequence during protein synthesis. What is this genetic code? So this is a system that is going to tell us about the amino acid sequence when there will be the protein synthesis. This is based on nucleotide sequence only. So now, till now what we got to know in this slide, this is the first slide of today's class. In this slide we got to know that replication and transcription, we know they both follow this complementarity rule, that purines is going to complement with pyrimidine. But that time they didn't know that how this complementarity exists between nucleotide and amino acid, like how this nucleic acid, chain of nucleic acid is going to make amino acid. But there was definitely this thing they have observed, that if there is any changes in nucleic acid, it is going to affect and influence what? Amino acid sequences in protein. So this concept led to the idea that okay, there is something what we call genetic code that dictates, that tells the like during protein synthesis, that what is going to be the amino acid sequence. Let's see. And this was based on nucleotide sequence also. So the genetic code, a challenging and multidisciplinary puzzle. So what's the first point? Determining the biochemical nature of genetic material and DNA structure was exciting. Proposing and deciphering the genetic code was exceptionally challenging. This effort required collaboration from scientists across various disciplines, like physicists, organic chemists, biochemists, geneticists. So in this slide what they are telling, that when the world got to know about the biochemical nature of the genetic material, that DNA is the biochemical, that DNA is the thing where this all genetic information gets stored. Okay, this biochemical nature of genetic material, when people got to know, then the DNA structure, the double helix structure, when people got to know, it was very exciting. But proposing and decoding this genetic code was exceptionally challenging. And this effort is going to require collaboration from many scientists across various disciplines. It's not just only about biologists, it's about different, scientists from different various disciplines, like physicists, okay, who are physicists who study physics, then organic chemists who study organic chemistry, the biochemist who study biochemistry, and the geneticist who study genetics. All from this all various disciplines, scientists are needed to decode this genetic code. It was exceptionally challenging. So now we will study about the key contributors of this genetic code encoding, who decipher this genetic codes, help in deciphering this genetic code. So let's start. First is George Gamow. He was a physicist. Okay. He proposed a key idea. What was the idea? The idea was triplet code. Now what is this triplet code that we will get to know? So in this we can say, so here, okay, so from here you guys just listen it as a story, okay, and try to understand like how they came to this conclusion of triple code and all those things. So now understand each and every point. So what was this? That only 4 bases exist, yet they must code for 20 amino acids. So before everything, you guys need to know that there are on only 20 amino acid, okay, that are used by the living organisms. The only 20 amino acids, 20 different amino acids that are found commonly in proteins. While there are over 300 known amino acids, only these 20 are used by living organisms to build proteins. Okay, these 20 amino acids can be arranged in countless combinations to create a vast array of proteins with diverse function. So you guys got to know that how much protein amino acids that are going to be there. Only 20 different amino acids that are going to be present in living organism. Means living organism use only these 20 amino acids to build it proteins. So the thing was like that there are only 4 base exist. We know that the nucleotide has something to do with these things, okay, and they are saying that there are only 4 base exist. Here we will talk about mRNA, okay, so it will be like adenine, uracil, cytosine and guanine. Only 4 base exist, yet they must code how many amino acid? 20 amino acid, because we know that living organism need these 20 amino acid to build different proteins, okay. So what was the conclusion? That the code must therefore involve combination of the bases. It means that it's not like that one base is going to code for one amino acid. We know that there are only 4 base and it is encoding for 20 amino acid. Means there will be an involvement of combination of bases that are going to code for these 20 amino acids. So then what he did, he suggested a triplet code, 3 nucleotide per codon. This was significant proposition. So now, okay, you guys can ask why triplet code he has proposed. He could have proposed 1, he could have proposed 2. So here you guys have to understand. So this was the very clear that 4 bases exist. Okay, if one base is encoding one amino acid, then it means this 4 bases is going to code for 4 amino acid only, and that is not possible. We need 20 amino acid. Okay, if you will say that there will be only 2 nucleotide that is doing what, that is coding for an amino acid, that is also not possible, because if there will be only 2 nucleotide, then it will be like 4 into 4, and 4 into 4 is what? It is 16. We also know that 16 is less than what? 20 amino acid. But if you will see a triplet code, it will be what? 4 raised to power 3. Then it will like 4 into 4 into 4, that is what? 64 codons. Means this 3 nucleotide per codon. I know they know that it was like very surplus, because we need 20 amino acid and there are 64 codon. But later we got to know that you will understand later. But here you guys need to understand that what he proposed that there will be combination of bases, and he suggested what? A triplet code, means 3 nucleotide per codon. And this would generate how many codons? 64 codons. Why? Because it will be like 4 into 4 into 4, and we know that 4 into 4 into 4 is what? 64. This creates a surplus of codons compared to the 20 amino acid that needed to be encoded. Means we are saying that we needed only 20 amino acid in living organism to build what? To build all the kind of proteins. So this 3 combination, 4 into 4 into 4, it's going to generate what? 64 codon. Means it is surpassing it. It's a lot of codons is generated as compared to the only 20 amino acid that needed to be encoded. But later we will see it has different features, properties. Then experimental proof came. Who gave the experimental proof? It is Hargobind Khurana. So he developed a chemical method, Okay, he did what? He synthesized RNA molecules with defined base combination, that is homopolymers and comonomers. So he synthesized what? RNA molecules which has what? Which has defined base combination. The first one he made was a strand repeating the two nucleotide. Means the first RNA molecules which he made, it was like this. It is repeating this two nucleotide only. This uracil, cytosine, uracil, cytosine, uracil, cytosine. Here's the picture of Hargobind Khurana. Then Marshall Nirenberg, what he did, he developed cell-free system for protein synthesis. Okay. This helped in decoding what? Genetic code. So I am not going into very deep. You guys just know these important terms like what they had did or what they hadn't. Like if talk about the Hargobind Khurana, what he did in this, he has made the RNA molecules with this repeating nucleotides, that is uracil and cytosine. And then comes this Marshall Nirenberg. What he did, so he developed a cell-free system for protein synthesis, and this helped in what? It helped in decoding the genetic code. Then who is he? He's okay. So next experiment, Severo Ochoa. What he discovered? He discovered polynucleotide phosphorylase enzyme that enabled what? RNA synthesis with defined sequences. It achieved template independent enzymatic RNA synthesis. So actually what we are here talking about, so where we have started, so we have started with Hargobind Khurana. So he has a key contribution in what? In this genetic code. Okay, he like able to do this contribution to the genetic code. How? By chemically synthesizing what? RNA molecules with repeating nucleotide sequences and then observing which amino acid were incorporated into proteins during translation. This research work, along with that of Marshall Nirenberg, which we have studied, okay, led to the cracking of this genetic code, and even they shared Nobel Prize. Okay. Then came this proof, which is Severo Ochoa. What he did, he discovered the polynucleotide phosphorylase enzyme, which enabled what? Which enabled the synthesis of the RNA with defined sequences. Okay. And by this, what they achieved? So they achieved the template independent enzymatic RNA synthesis. They are able to synthesize RNA, okay, which is template independent. So now what came? This is the contribution that all the scientists have gave. You have to just remember the key points. So here is the checkerboard for genetic code. Now here you guys have to understand more carefully that these combined efforts led to the creation of what? A checkerboard representation of the genetic code. So here you guys can see what it is. So the 4 nucleotide bases, that is U, C, A, G, that uracil, cytosine, adenine and guanine. Here again U, C, A, G. This is the first position, this is the second position, and the third position here it is like UCAG, UCAG, UCAG. So how they have written this checkerboard? So for first position they have took this U, second they have took this U, and third position they have take this U. Then for this they have taken U, U, C. Just like this they have made this checkerboard, which is going to tell like different amino acid that has been formed from this nucleotide base pairs. Now what are the salient feature of this genetic code? So if you will try to understand what was the genetic code, salient feature of this genetic code, first of all they are triplet in nature, means sequence of 3 nucleotide, what we call a codon, codes for a single amino acid. They are total how many codons? We know it is 4 into 4 into 4, that is 64 codons present. 61 codon specify amino acid, whereas 3 codons don't code for any amino acid, but it serve as stop codon. So what we are telling, the first salient feature of this genetic code is that this is a sequence of what? 3 nucleotide, what we call together as a codon, and this is going to code for a single amino acid. We have studied that there are 64 codon present, in which 61 only going to specify or code for amino acid, whereas 3 codon will not code for any amino acid, but if they are present, it means now stop the translation. It means it is acting as stop codons. Second feature, this was the triplet nature. Second feature is degeneracy. If you will talk about degeneracy, what is degeneracy? That multiple codons can code for the same amino acid, and this is why even we need 20 amino acid, they are present of 64 codons. Okay, so that's why I told you that we will answer the question why they have surpassed this number, that if we only needed 20 amino acid, why there is presence of 64 codons, because multiple codons can code for same amino acid. And or what we can say it in another way, that some amino acid are coded by more than one codon. These both statements mean same only, and this is what defines the degeneracy of the genetic code, that multiple codons can code for same amino acid. This redundancy is a key characteristic. So for example, you guys can see that leucine, okay, leucine can be encoded by 6 codon, UUA, UUG, CUU, CUC, CUC, CUA, CUG. So we can see that 6 codon can code for only one amino acid, that is leucine. And that's the reason why we have said that even we need only 20 amino acid, there is presence of 64 codons. Why? Because same multiple codons can code for same amino acid. This is one of the key characteristic of genetic code. The first we have read the triplet nature, then we have read degeneracy. Now the third salient feature which came into the picture is the continuous reading. Codons are read continuously without gap in the mRNA. There are no punctuation marks between codon. The codons are sequentially and non-overlapping reading. So what does it mean? So let's see. Okay, here is the example. You guys need to understand it, see it properly. That we are saying that codons are read continuously without gaps in mRNA. Means if we are this is an mRNA, okay, how we will read the codon? It will be like AUG. This is the start codon, which we are going to talk about more in upcoming slides. AUG, one codon, CAU, GAC, UCG, and UGA. Okay, this is how we read a codon. We read it continuously. We didn't give any gap in mRNA. There was no overlapping. It is not we are saying AUG and GCA. No, it is AUG, one codon. Then we will read it like CAU, second codon. It will be like this. Okay, there should be no punctuation mark between codon. It can't be like AUG dash CAU. No, it won't work. There would not be any comma, any punctuation mark, and these codons are read sequentially and non-overlapping. I have given you the example. Then the fourth features came, that it is universal in nature. What does it mean? That code is largely similar across organism. That means one codon codes for same amino acid in different organisms. Example, UUU codes for phenylalanine in most organisms. Exceptions are mitochondrial codons and some protozoans codon. So again, here you have to understand there is difference between degeneracy and universal nature. What was degeneracy means? Degeneracy means that multiple codons can code for one amino acid. Okay. But what is this universal nature? Universal nature means is that if UUU is coding for phenylalanine, So it is going to code for phenylalanine in most of the organism. Okay, there are some exception like mitochondrial codons or some protozoans codon, but most of the organism we will see that the UUU codon is coding for phenylalanine only. So this is the universal nature. Then now AUGs, I have told you that I will tell you about the start codon. So what is the role of AUG codon? So it has dual role. AUG is going to code for which amino acid? It is going to code for methionine amino acid. As well as this codon AUG will act as what? Will function as what? As start and initiator codon. So if we wanted to start the translation, you should have this AUG codon. This AUG codon will tell, okay, now start, this is the initiator codon. Now, if there is some start codon, then there will be stop codon. And we have studied in few previous slide, we have seen that there are 64 codon in which 61 is coding for amino acid, whereas 3 is not coding for anything, but acting as the stop codon, which is going to stop the translation process. So these 3 stop codons exist. It is UAA, UAG and UGA. Okay. This is also people called UA, people call ochre. Then this UA ochre, UAG people say amber, and UGA people say opal. Ochre, amber or opal. These are the that you guys don't need to remember. You guys just need to remember the stop codon, that UA, UAG and UGA. These are what? These are the stop codon. If they are there, it means now translation has to stop. These are the terminator codon. mRNA signals to stop making protein. UGA, UAA and UAG. These are the 3 stop codons. We have one start codon, that is AUG, and then 3 stop codon, that is UAA, UAG and UGA. So we have studied about this genetic code. We have studied a salient feature. Now let's come to what? Come to mutation and genetic code. So now will understand what? We will understand this gene DNA relationships through what? Through mutation slide studies. What is a mutation? So mutation is nothing but a change in the DNA sequence. If there is any change in the DNA sequence, we say, okay, the sequence is being mutated. Mutation has taken place. If we talk about the large scale mutations, so large deletion or rearrangement in DNA segment have noticeable effect. So what are these large scale mutations? So if in the DNA sequence, there is some large deletion, okay, there has been deletion of 3-4 nucleotide together, or there is a rearrangement in DNA segment, like 3 nucleotide come before and after, all those things. So this is going to have what? It is going to have noticeable effect. Why? Now I think you guys could understand why this is happening, because we are saying this mRNA, this is triplet code, okay, and this codon is going to code for amino acid. So you just think, if these mRNA is translating, okay, and this mRNA strand this segment got mutated, or DNA segment got mutated, what will happen? Now it will make different codons. If you are making different codon, it will going to code for different proteins, and this is going to alter the normal function. These mutation led to what? These mutation led to loss or gain of entire gene, thereby affecting what? The specific function that the gene was playing. This was the large scale mutation. If we talk about the point mutation, what is a point mutation? So point mutation is a change in a single base pair. Example is sickle cell anemia. A single base substitution in the gene for beta globulin chain result in the change of amino acid. Normal GAG codes for glutamic acid, whereas mutated GUG is going to code for valine. So what is a point mutation? So a point mutation is what? So point mutation is if there is a single base pair change, okay, single nucleotide change in the DNA sequence, it is what we call point mutation. What is the example of point mutation? So here we can see the sickle cell anemia. So what happened in this sickle cell anemia? So usually our RBC is of normal this shape, okay? And in this case of sickle cell anemia, what happened? This RBC becomes sickled in shape. Then there will be, there won't be space. Then there will be less hemoglobin or no hemoglobin. So the oxygen transfer is going to be like, very difficult, and that person is going to suffer from sickle cell anemia. So why it happens? It happens just because of this point mutation. What happens actually in this? So the gene which is going to code for this beta globin chain, there is a point mutation in that gene. Okay, so if we're talking about the point mutation, so if you guys can see what happened, it's just a single mutation, you understand? In normal there was this GAG, and this codon was coding for what? Glutamic acid. But mutation, what happened? This A got substituted by uracil, and what it became? GUG. And now this GUG is coding for what? It is coding for valine amino acid. And just because of this single mutation, the normal RBC became sickle cell, and that's how people suffer from sickle cell anemia. The one change and the person is going to be affected. So here we can see that how a point mutation has led a person to become what? Sickle cell anemic. Now after this point mutation, let's talk about this frame shift mutation. What is the definition of the frame shift mutation? It is the insertion or deletion of one or two bases. This alters the reading frame during protein synthesis. What are their effects? This changes the reading frame from point of mutation onwards. This results in a completely different and usually non-functional protein. So in this slide, what they are saying, we have studied this point mutation where just a single base pair is going to be substituted or something like that. But when we talk about frame shift mutation, what happens in this? There will be insertion or deletion of one or two bases. Now what will happen? Because there has been either deletion or either insertion of 1-2 bases, now the reading frame, the frame which we are reading, 3 codons we are reading now, it get altered. How? And because of that, it is going to result in completely different and usually non-functional protein. So how it gonna happen? That here is also one example in figure it is shown, but in later slide it has given more better examples. Let's see. First we will understand it with a basic example, then we understand this figured example. So for example, a sentence composed of 3-letter words is used to represent what? Genetic code codon. So let's see. Just like we know now that there is only 3, this codon is of what? This triplet in nature. So we have written a sentence, okay, with 3-letter words only, and you just think that this is doing what? This is representing our genetic code. So what is this? Ram has red cap. Okay. So now we will see the example. For example, if we have inserted one base in here, like we have insertion of B in between has and red. So now how would this rearrangement will happen? We have inserted B where? We have inserted B here. So now we will try to break it into 3. How it will be like? Ram, it will be fine. Has, it will be fine. But now because it has B, so it will be like Bree, DCA and P. Does it making any sense? No. Just like that, if we will delete one base, for example, we will delete R from here, what it will be like rearranged? Ram will be fine, has will be fine, but because we have deleted this R, it will be like E, D, C and AP. Does it making sense? No. So like this only codon also work, genetic code also work. If there is any insertion or deletion, it is going to do what? Frame shift mutation. Now let's see the example. If we insert 2 bases, if we are going to insert two bases, for example, we are inserting bi in between has and red. So you guys now can see what it is. Like ram will be like fine, ram has bir edcap. Does it making sense? No. Let's delete two bases. If we will delete this re, what it will be like? What will be the rearranged statement will be like ram has dcap. Now again, is it making any sense? No. Just like this, if there will be any insertion and deletion of base pairs, nucleotides in the genetic code, it will be do the same. And this alphabet represents what? These alphabets is nothing, it is analogy that, yeah, it is like base pair, like nucleotide only. Like for example, we have seen now like AUC, GUC, just like that. It is, we are analogy that with it. Okay, all of this change cause what? It cause frame shift mutation. Changes the reading frame from the point of insertion or deletion onward. The point where we have inserted or deleted any base pairs, it is going to change the reading frame. Let's see now. If now we can think like, okay, what if we will delete or insert three or multiple of three bases? This mutation lead to what? Now it here what will happen? It is not like that. It is not going to make any sense, because now this time it is 3, but it is going to do what? This mutation leads to the insertion or deletion of one or more what? Codons. This result in what? In insertion or deletion of one or more what? Amino acid and protein. And reading frame remain unaltered from that point onward. Changes in 3 bases, adds, removes amino acids. So for example, let's see this example. Okay, so like for example, this is the DNA sequence. Okay, here we are taking DNA sequence. We know that what we are going to be like, DNA is going to change to mRNA, but you know, we are talking about the mutation and that happens in DNA only. So you just leave that step. You just think that this mRNA step will be like in between. So for example, these 3 codons were making some amino acid, and because of this amino acid, this protein will be like formed. This polypeptide will be formed. But now you just think, if there will be insertion of 3 base pair codon, what will happen? This 3 base pair codon is going to code for amino acid. But now what will happen? This sequence will be modified. So it may happen that before it is coding for some protein, now it is coding from some different protein that is not even maybe useful or something like that. That may be some, you know, harmful protein. Don't know. But this amino acid sequence is going to be what? Is going to be modified. Now if we will try to, you know, see the example using that or this sentence letters word only. So now what will happen? If we are trying to insert big in between has red, it will be like Ram has big red cap. So it's not like that there is something came which has no meaning. It has meaning, but it has changed the sentence from the previous one. Before, Ram has only red cap, but now Ram has big red cap. Okay, and even if you talk about deletion of 3 base, for example, we are deleting red statement. So what it will be like? Now it will be like, previously we were telling that Ram has red cap. We were specifying that Ram has what? It has he has a cap which is red in color. But now they are specifying nothing. They are just saying that Ram has cap. It is making sense. It is not like it is not making sense. But this is not same from the previous sentence. So this was all about genetic code and mutations. Now we will move to what? We will move to tRNA, that is the adapter molecule, and this is from NCERT 5.6.2 section. And this is basically what this here what we will study. Here we will study about the structure of transfer RNA and function of the transfer RNA. We are talking about this tRNA from like, you know, from last 2-3 classes, but today we will understand it more clearly. So first we will study about what? First we will study about Francis Crick adapter molecule hypothesis and tRNA. amino acid lack inherent features to directly recognize and bind to the genetic code. Francis Crick understood the need for a mechanism to read the genetic code and connect it to specific amino acid. Adapter molecule hypothesis then came. So what does it going to mean and what is meaning actually? So amino acid don't have any feature which is going to directly recognize that okay, so this is my genetic code and I will go and I will bind to this genetic code. No, it don't have any inherent feature like that. Okay. Therefore, Francis Crick, the scientist, he understood that there is a need for what? There is a need for the mechanism in which, which is doing what? Which is reading the genetic code and connecting it to specific amino acid. Means that particular mechanism, that particular thing is reading the genetic code and then it is connecting it to the specific amino acid. So there is some mechanism in between and that need to be, to be understood. So he then gave this adapter molecule hypothesis. So what happened in this adapter molecule hypothesis? Crick proposed the existence of an adapter molecule with a dual function. So Crick proposed that there is an existence of an adapter molecule and it has two functions. First, it reads the genetic code and second, it binds specifically to the individual amino acid. Because we know that there is no amino acid. No amino acid means amino acid has no any inherent features that is going to recognize and bind to the genetic code. Okay, so Francis Crick did what? He said, okay, there is some molecule, okay, he named it as adapter molecule and it has two functions. First, it is reading the genetic code. It is reading the codon and then binding specifically to whom? To the individual amino acid for which that genetic code is for. Then this tRNA, the transfer RNA, given to picture a molecule that could read the code on mRNA. Means this molecule can be able to do what? It will be able to, able to read the code on mRNA and then it can also bind to a specific amino acid for which that codon is coding. Okay. tRNA, initially called sRNA or soluble RNA, it was already known before the genetic code was fully understood. tRNA's role as the adapter molecule was later recognized. So what is in this slide they are saying? Now Francis Crick said that there is some adapter molecule, okay, and which has two functions. First, first that molecule is reading for what? That molecule is reading for the genetic code. Okay, it reads the genetic code, and second, it go and attach to that specific amino acid. But at that time, they didn't know that it is tRNA only. They know this tRNA. They know there is this tRNA, but with the name of sRNA, that is soluble RNA. But when they got to know about this genetic code properly, then they understood this tRNA's role as the adapter molecule. So here you guys can see this yellow color is what? This yellow color is tRNA. So this tRNA is doing what? So this tRNA is reading the code. And how it is doing that and although that we will study later as in the upcoming slides. So this code and it is also attaching it to the specific amino acid. So this is how it works. Now if we will talk about the structure of tRNA, transfer RNA, if we talk about its 2D structure, it resembles a clover leaf. If we talk about its 3D structure, it looks like an inverted L. So here is a picture. If we talk about its 2D, that is two-dimensional structure, it is looking like what? A clover leaf. Here you guys can see. Okay, it's anticodon loop, we will understand later. And if we talk about this three-dimensional, it is looking like this inverted L, where it is here anticodon part, and this is what? This is the amino acid to which it is connected. So we are talking here about the key features of the structure of tRNA, and from here you guys can answer the NCERT exercise 8B. So if we talk about the structure of the tRNA, it has some key features, that is anticodon loop. It has bases that are complementary to a specific mRNA codon. This loop binds to the specific codon according to the base complementarity. This is how it reads the code. So now what? So this tRNA has this anticodon loop. Okay. And now this anticodon loop has what? This anticodon loop has bases, okay, has nucleotides that are complementary to what? That are complementary to the specific mRNA codon. For example, this codon is AGU, okay. So now this tRNA is going to have anticodon, which is what? In which this tRNA is going to have the bases that are complementary to this codon. That will be like UCA. That's why we say it as anticodon loop, because it is going to have bases which is complementary to what? To the codon. And this is how it reads the genetic code. Then we talk about how we it reads the genetic code. Then the amino acid receptor end, the 3' end of the tRNA called acceptor arm, it has CCA sequence. The corresponding amino acid binds here. So here we studied that here is like what? It has an anticodon loop. It has the basis which is complementary to what? To the basis which is present on the codon. It has the amino acid receptor end at the 3' end. It has amino acid receptor end. This is what we called as, it is called as receptor arm. Okay, and the corresponding amino acid binds here. Like for whatever this codon is coding for which amino acid it is coding for. So this end is going to have like corresponding amino acid binds here. Now, if we talk about the structure of tRNA specificity, so for each amino acid there is a specific tRNA. Initiator tRNA. So for a specific tRNA, we said for each amino acid there will be a specific tRNA. So there will be a specific tRNA for what? For this start codon AUG. There are no tRNAs for stop codons. So here we have seen that initiator tRNA. This initiator tRNA will have this anticodon which have basis what? UAC, which is complementary to this codon, start codon, that is AUG. And here on this acceptor arm, it will be like have what? It will have methionine amino acid. But there is no tRNA for what? For stop codons. So now what is summary of today's class? First, we have set of rules which is translating what? DNA RNA sequence into proteins. What roles are like? This genetic code has like what features? It is universal, it is non-overlapping triplet codons, 64 possible codons which is coding for 20 amino acid. Multiple codons can code for same amino acid. If we talk about mutations, there are changes in DNA sequence, for example, point, frame shift, insertion, deletion. It can be harmful, it can be beneficial, it can be neutral. Causes for this mutation will be like radiation, some chemicals, error in replication and all. If we talk about tRNA, that is the transfer RNA, it is adapter molecule which is linking codon to amino acid, to its respective amino acid, contain anticodon and amino acid attachment site, help in protein synthesis during translation, has clover leaf structure. If you will see from 2D structure, it has clover leaf structure. Now there are some NCERT based questions. Let's see the first questions. First question says that which of the following is not a characteristic of genetic code as described in the passage? So they are asking like which among this option is not a characteristic of the genetic code. The code is degenerate, the code is punctuated, the code is nearly universal, or code is triplet. So we know the answer is second, that the code is punctuated. The genetic code is continuous and does not have any punctuation mark like comma or periods that separate the codon. Degenerate, they are degenerate means multiple codons can code for same amino acid, making the code redundant. Nearly universal means most organisms use the same codon to code for the same amino acid with only a few minor exceptions. They are present in triplet. A codon consists of how many nucleotides? 3 nucleotides. The next question, What is the dual function of AUG codon? We know that what is AUG codon? It is a start codon. It codes for phenylalanine and act as a stop codon? No. It codes for methionine and act as a stop codon. It do code for methionine, but it is not acting as stop codon. Codes for methionine and act as initiator codon. So yeah, we know that it is right, that it is code for methionine and it act as what? Initiator, starting codon. Whereas we know that it is not coding for phenylalanine. Option is 3, that AUG has dual function. It codes for methionine and it acts as initiator codon. Question number 3, that which of the following is a stop codon? Is it AUG? No, we know that it is what? It is start codon. UUU, we know it is coding for phenylalanine, so no, it is not a stop codon. But UA, okay, this is stop codon. So answer is C, UA. UA, UAG and UGA are the 3 stop codon in the genetic code, signaling the end of protein synthesis. AUG is the start codon which initiate protein synthesis by coding for which amino acid? Methionine. UUU codes for which amino acid? As I said, phenylalanine. And GGG codes for which amino acid? Glycine. Next question, who postulated the presence of an adapter molecule that would on one hand read the code and on another hand would bind to specific amino acid? So right now only we have studied, it is Francis Crick who has given what? This adapter molecule hypothesis. It was Francis Crick. He proposed the concept of transfer RNA as the adapter molecule in protein synthesis. It reads the codon on mRNA and brings the corresponding amino acid to ribosome. Nirenberg is known for his work in deciphering what? The genetic code, specifically identifying the codons for specific amino acid. Khorana contributed significantly to understanding the genetic code and confirmed the triplet nature of the codons. Gamow proposed the concept of genetic codon, that it consists of 3-letter codons, but he did not specifically suggest the adapter molecule. Last question, that which enzyme was helpful in polymerizing RNA with defined sequence in a template independent manner? Is it DNA polymerase? It is RNA polymerase? Is it Severo-Ochoa enzyme, that is polynucleotide phosphorylase, or it is reverse transcriptase? So here we know it is Severo-Ochoa enzyme, which is a polynucleotide phosphorylase. So what it does, it is also this polynucleotide phosphorylase, it is also known as Severo-Ochoa enzyme. It can synthesize RNA molecule with specific defined sequence without the need for a DNA template. But we know that what is DNA polymerase? It is an enzyme which is primarily responsible for what? For replicating DNA, not synthesizing RNA. Then RNA polymerase, we know it polymerase what? Synthesize RNA. It always requires a DNA template to guide the sequence of new RNA molecule. Whereas what is reverse transcriptase? It is an enzyme used by retrovirus to convert the RNA genome into DNA, not to synthesize RNA from scratch. So what are today's keynote? That genetic code, this is universal across most organisms, uses 64 possible codons. Start codon is AUG and has 3 stop codons. Reductant degenerate means multiple codons can code for same amino acid. Then we studied a mutation. It can be beneficial, harmful or neutral. Point mutation affects single nucleotide, whereas frame shift mutation alter the reading frame. UV radiation chemicals can cause mutation. If we talk about tRNA, it is shaped like clover leaf, has anticodon loop, carries amino acid, act as translator between mRNA and protein. So here are some interesting questions that you guys need to answer. That first, in a molecular biology lab, a student observed that a bacterial cell was simultaneously transcribing and translating a gene, while a eukaryotic cell couldn't do the same. Explain why this difference exists between prokaryotic and eukaryote. So this is about this polyribosome and polysome that you guys can answer. Second question, if RNA polymerase 2 in eukaryote is damaged, which type of RNA would still be produced normally and why? So we know that this eukaryotic has 3 types of polymerase. Okay, so we have to answer if first and 2 is proper, then which is going to be produced. I'm giving you hints that you can Answer. Okay. Third, a genetic disorder result from failure to remove introns from pre-mRNA. Would this condition be more likely to occur in bacteria or human? Justify your answer. So, a genetic disorder can be resolved from what? Failure to remove intron from pre-mRNA, because we know that through splicing we remove this intron. So would this condition be more likely to occur in bacteria or in humans? So you have to answer. We know the splicing step occur in which? Prokaryote or eukaryote? So you guys can answer this also. So now let's see the first question. First question is, which of the following is a stop codon in the genetic code? So we know that there are 3 stop codon in the genetic code: UAG, UAA and UGA. So here we can see which option is there: UAG. Whereas we know that this AUG is what? AUG is the start codon. So answer is UAG. Next question says that the genetic code is considered degenerate because we have studied one of the feature of this genetic code, that it is degenerate, means multiple codons can code for single amino acid. So let's see which option is fitting this. One codon codes for multiple amino acid? No. Multiple codons can code for same amino acid? Yes. Codons are read without punctuation? Yeah, that is correct, but this is not about degeneration. The code is nearly universal across organism. That is also true, but it's not about degeneracy. Okay. Then third question, that a frameshift mutation occurs when? It occurs when there will be 1 or 2 nucleotides are inserted or deleted, disrupting what? The reading frame. So the answer is option number 3. Then which scientist contributed to deciphering the genetic code by developing a cell-free protein synthesis system? So we have studied that who has discovered this, developed a cell-free protein synthesis system which helped in deciphering this genetic code. It was Marshall Nirenberg. Whereas George Gamow was the physicist who gave this idea of triplet code, and this Har Gobind Khurana, what he gave, so he told us about he synthesized that RNA molecule with repeated nucleotide UG, and Chevreau-Shah, that enzyme thing. Okay, so the scientists who contributing to deciphering the genetic code by developing a cell-free protein synthesis system is Marshall Nirenberg. The anticodon loop in tRNA is responsible for what? It recognizes that anticodon loop in tRNA is responsible for what? It is binding to amino acid? No, we know that the acceptor arms do that. Okay, it is initiating the translation? No, we know that AUG codon does that. It is terminating the protein synthesis? No, we know that the stop codon is responsible for that. It is doing what? It is recognizing the complementary codon on mRNA. So let's save it, submit. Yes, submit, and here we can see all options are correct. So it is all about today's class. So bye, we will meet tomorrow. Thank you.